welcome. Thank you for coming to this session on brain development and aging. I am Jean Chen from the University of Toronto, and this is my co-chair, Tang Xu from Child Mind Institute in New York. So we're going to start with the first talk entitled Development of Segregation and Integration of Functional Connectomes during the first 1,000 days. And the speaker is Qiongling Li from Beijing Normal University.
Thanks for the introduction. And hello, everyone. Today, I'm going to talk about the development of segregation and the integration of functional connectors during the first 1,000 days. Okay, I have nothing to disclose. To start with, let's first understand why studying the functional connectors during early life is crucial. The first 1,000 days from conception to the first 3 postnatal years are critical period in the human lifespan. It undergoes a remarkable growth and reorganization during this period, laying the foundation for future human brain development and later cognitive growth. As illustrated in the blue box, during this period, the human brain undergoes a series of rapid and complex cellular and molecular processes, which significantly shape the architecture of the cerebral cortex. Serious researchers have documented that the white matter tracks and the networks is largely established, while the intrinsic or spontaneous functional network develop rapidly after birth. The functional segregation and integration refer to the key organizational principles, which refer to the brain regions become specialized for specific functions, and these specialized regions work together as a cohesive network to support more complex processing. The continuous longitudinal development of functional segregation and integration across the whole brain connectome remains, during the first 1,000 days, remains largely unexplored. Thus, we explore the age-related changes of functional connectivity strength, functional segregation and integration, their implications on later neurocognitive growth, as well as the underlying molecular mechanisms underpin these processes. First, I will introduce the datasets we used. We used 2 public available datasets, the Developing Human Connectome Project, Baby Connectome Project. We conduct a comprehensive quality control and data cleaning process for the images. In total, the 2 combined datasets include 665 infants with 930 fMRI scans. We used the generalized addictive mix model to capture the developmental trajectories. The FCS, the functional connectivity strength, is one of the most direct measures for the overall functional connectivity. We investigate the age-related changes in FCS. As we can see here, the significant age-related changes in FCS, mainly in the sensory motor, visual, auditory, and also regions within default mode systems. To illustrate the different developmental patterns of the FCS, we categorized the nodes with significant changed FCS into 4 clusters. We then calculate the functional connectivity between each cluster and all voxels. We found that the significant changed FCS cluster-based FCS mainly within each functional system. The F ratio to the total F values, the ratio of F values to the total F values in each functional system, as shown in the radar plot, support this finding. HAPs were identified as FCS greater than greater than the mean plus 1.5 times standard deviation. the hub distribution across different functional systems at different ages was shown in the upper plot. It's clear that the hub mainly in the perinatal period, the hub mainly in the sensory motor system, and in the postnatal years, the hub was mainly in the dorsal attention and the visual systems, then transitioned to the default mode and potentially undergoes a reorganization at the 3 years of age. The hub voxel of the sensory motor system initially decreased from a relatively high level and then increased to some extent. The hub voxel of the dorsal attention system first increased and then decreased. For the default mode and the visual system, they continue to increase with the default mode have higher voxel ratio. Then to explore the spatial extent of the FCS, we computed the FCS, we investigated the age-related changes of FCS across distance. We found that the short-range FCS showed significant changes across the extensive brain cortex, cerebral cortex. While for the medium to long-range FCS, only a few regions observed a significant change. The overall of the FCS across distance beams all showed an inverted U-shaped trajectory. It's interesting that the peak age across different distance beams showed were different, and with the short to medium range FC connectivities peak earlier than the long-range connectivities. We then investigate the development of functional segregation and the integration. The clustering coefficient, a measure of the functional segregation, showed quantifies the extent to which the node communicate with each other in a graph. It shows an inverted U-shaped developmental curve. Then we investigate the functional integration by analyzing the nodal efficiency, which quantifies the efficiency of a node communicates with other nodes. It showed a U-shaped developmental trajectory. To explore the progression of the segregation, the functional segregation and the integration development across the cerebral cortex, we used the principal component analysis to capture the differentiation developmental trajectory across the cortex. We found the first component we regarded as the principal developmental axis. We found that the principal developmental axis differentiate from the anterior to posterior, which resembles the variation of functional connectivity profile along the anterior to posterior, which is considered as an immature sensory motor to association axis. Then we investigate, our hypothesis is that the neonatal functional connectors could underlie the later neurocognitive development. So we use the support vector regression method to predict the neurocognitive outcomes at 1.5 years age, and from predictors at birth. We found that the mean FCS nodal efficiency modularity could significantly predict the cognitive scores, and the FCS clustering coefficient nodal efficiency modularity within module functional connectivity could significantly predict the language scores. However, for the voxel level analysis, it didn't yield a significant prediction. Lastly, we explored the underlying molecular mechanisms underlying these processes. The FCS is the basis of functional segregation and integration. We then use the developmental gene dataset, the brain span atlas, to compare the transcriptional level between regions with significant FCS changes and non-significant FCS changes. we found that we performed the gene ontology expression enrichment and gene set enrichment analysis. We found that the functional connectome development is regulated by the typical neurodevelopmental processes, including dendrite development, synapse development, neurodifferentiation, neuromigration, axon development, and myelination, as well as genes involved in metabolic processes, including aerobic glycolysis and oxidative phosphorylation. So to summarize, our take-home messages are brain hubs shift from primary to association regions during the first 1,000 days of life. And the function segregation and integration develop along anterior to posterior axes. Neonatal outcomes could predict the cognitive and language outcomes at 1.5 years of age. Network growth is regulated by genes involved in neurodevelopment and metabolisms. And I would like to thank Dr. Yonghe's supervision and Dr. Li's support and all our collaborators. There is a lot of details I have not described here. If you are interested in, you can email me and or meet me at the poster. Thanks all for your attention.
Thank you for that wonderful talk. Thank you for that wonderful talk. Any questions? Yes?
If you compare the cluster function and the efficiency with adults, on average, how is it different with the babies?
Okay, sorry. Thank you for your question. We didn't compare the clustering coefficient between the babies and the adults.
Okay, there's a question on the app. How is the intra-subject reliability in infants compared to adults?
Sorry, can you repeat it?
The within-subject reliability of the measurements, did you assess that?
No.
And what genetic data was used? Was it available through the Baby Connectome project?
It's from the brain span atlas. It's a developmental expression. It's a developmental gene expression data set.
Developmental gene expression data.
We only included the participant aged between 28 postnatal weeks and 3 years.
Okay, thank you. I thank our speaker again.